Good morning, everybody. My name is Lieven van Vooren from the company Pharmavize, and my talk of today is titled Appropriate Standards and Practices in Early Drug Development and Manufacturing. So here you have the table of contents of my presentation. So I will start briefly with a short company profile. Then I will tell a little bit about chemical pharmaceutical development, being formulation development and analytical development. Then a few words about GMP manufacturing of early clinical trials, and also a small case study giving you a project plan, how we handle such project. And last, I will end up with some take-home messages. So Pharmavize is a company that has been founded in 1995, so almost 20 years, and we operate from Mariakerke near Ghent in Belgium. So we are a service provider, so we provide CMC services to the pharmaceutical industry. So we do not have our own product development, we only work for pharmaceutical customers. So we are today with about 45 experienced people, and our activities have been expanded over the last year. So we started as a regulatory consultancy company in 1995. Then we went into contract research activities around 2000, and in 2006 we also obtained a GMP license for the manufacturing of clinical trial, non-sterile clinical trial materials. So since 2006 we have this GMP license. So in fact Pharmavize has two different business units. So the left-hand side unit is what we call the Pharmavize services. So these are the, let's say, activities that we already do for a very long time, being CMC regulatory writing, technical writing, and also GMP quality management support like GMP audits or implementation of quality systems. And at the right-hand side, we have then the Pharmavize contract development and manufacturing organization. So this is the organization that will help pharmaceutical customers to bring their APIs in clinic. So a typical project executed at Pharmavize is when a customer comes with an API and asks us, please develop an appropriate formulation for a clinical study, first a phase 1 clinical study, and later also phase 2 and eventually phase 3 clinical studies. So in this unit, in this business unit, we have these activities. So the R&D activities are given here: formulation development, analytical development, and stability studies. And then the GMP activities, so GMP bulk small-scale manufacturing, labeling activities, and also import and release activities. So we serve about 70 customers. So the majority, about half of our turnover is for big pharmaceutical companies. Then we have the small and medium enterprises and the biotech companies, small companies, startup companies. So what are now the challenges in early drug development? Well, there are quite some challenges here. Note that my presentation is focused on small molecules, so the small chemical molecules, and the challenges today are, in the first place, the API properties. So more and more APIs belong to the classes 2 and 4. This means low bioavailability, low solubility, so they need a specific formulation approach. Compounds have more and more increasing lipophilicity, so log P values of more than 3 are commonly nowadays, meaning that also these need a specific formulation approach, increasing molecular weight, decreasing permeability. So this makes the APIs are for us more and more difficult, so more and more challenging to find appropriate formulations. The regulatory requirements become more and more stringent. So I will not go in detail here, but for a phase 1 study, you need to supply much more regulatory information than you should have done 10 years ago. And also competition is a challenge. Once a customer, a company has made a decision to go forward with a compound in the clinic, then everything should go fast. So there is a lot of competition, and once the client wants to go forward, we need to go in an accelerated development. Now, one of the first things we always discuss when we start a new project is what will be the strategy? Will we go for a liquid or will we go for a solid formulation? This is already a very basic question, and I give here some pros and cons for each of the two. So a liquid formulation has a very flexible dosing range. You can play with the concentration and the volume, so this allows a very broad range of doses. The development time is mostly faster than for a solid, because one of the points is that you do not have to take care about the solid state properties like particle size, polymorphism, salt screening. These activities are not needed or not fully relevant for a liquid formulation. But the disadvantage of a liquid formulation is that you might need a bioequivalent study when you want to switch the transition from a liquid to a solid, because most companies, they want to go in the later stages with a solid. So then if you start with a liquid, then at a certain moment during the development, phase 1, phase 2, you need to do a bioequivalent study to make the transition from the liquid to the solid. And that's exactly the reason that many of our projects start immediately with a solid. So immediately with a solid gives us an advantage that you can have seamless transition to later phases, but at the other hand, it will require more development work on the API because you have to take care about these solid state properties, and also requires typically more work for the drug product development. So here you can see for the early clinical trials, what do we now, and so it's a summary of, let's say, over the last years, what we have manufactured for early clinical materials. And you can see here that still a large quantity are aqueous solutions, aqueous suspensions, also including the nano suspensions. Then we have here also a liquid part, which is the non-aqueous solutions. For example, the API dissolved in an organic solvent like propylene glycol, polyethylene glycol. And then we have here the biggest block for the moment for Pharmavize manufacturing activity is the powder-filled capsule. So this means a simple formulation, the API blended with some excipients and then filled in a capsule. And then we have here an approach which is typically used for the low bioavailable drugs, that is dissolving them in a solid, in an oily vehicle, a lipid vehicle, and then subsequently fill this liquid vehicle into a capsule. So this has the benefit of a solid because it's a capsule, but also the benefit of a liquid because the API is dissolved in an oil. And then this part here, the blue one, these are then, let's say, more complicated formulations that we need in order to make the API more bioavailable, like, for example, spray drying, spray congealing, melt extrusion. They are here in this part. So we do not use it very often for phase 1, but if we use it, it means that we need it in order to have the API, to have the right bioavailability for the API. And last, this is the reconstitution formulations. When stability, for example, is an issue, then we have powder and liquid separate, and then we combine them at the moment of the clinical study. So this is here the technology that I just said for the lipid formulations. So the low bioavailable drugs, they require a special approach. So in this case, we will fill them or we will solubilize them in an oil. And so we have the benefits of a liquid formulation. It's applicable for a wide range of new chemical entities. So a lot of the new molecules fit in this principle. You have a lot of possibilities in the formulation work regarding the choice of oils, surfactants, combinations, co-surfactants, co-solvents. So a lot of combinations. Typically, we use also experimental designs in order to find the appropriate combination. And the technology is also available for commercial manufacturing. It's called LICAPS. It's a capsugel technology. So we have here the machine in-house to manufacture small batches, small quantities of this LICAPS technology for clinical studies. Now for the solids with a low bioavailability, we have other approaches that we apply. So this could be, for example, micronization, nanonization. So this is just reduce the particle size so that the surface, exposed surface is higher and the bioavailability, the absorption is better. Other techniques are spray drying, spray congealing. They both have as purpose to turn the crystallographic form into an amorphous form. So when the API is not in a crystal anymore, but in an amorphous particle, typically its bioavailability is also better. So this is the technique we do. We turn the crystallographic form into amorphous and then subsequently fill it in capsules after blending, for example, with a filler and a lubricant. Other technique, the same principle, melt extrusion. So we will dissolve the particles in a polymer. Also then, we don't have the crystallographic form anymore. The API is dissolved in the polymer, and then this can then be further formulated also in a capsule or a tablet afterwards. And other techniques we apply for these, let's say, difficult APIs, granulation, extrusion, spheronization, drying, coating. So this is also often used to have slow release formulations with targeted release profiles. We need to do additional activities as given here. So if we start a development project starting from the API and we need to develop a drug product, well, we will first check, evaluate some very important properties needed for the formulation development. These properties are the following, so some in vitro properties, so which is the solubility, the dissolution, and this is something that we have developed in-house, the resistance against precipitation, also called the IV2Sol platform. So I will give you a few slides about this platform. Other attributes that we measure is the intrinsic stability, and so we will set up stress testings on the API to see what are the, let's say, the stress conditions degrading the API. For example, if oxygen is an issue, then we need to know that we need to formulate, for example, with an antioxidant or with a specific packaging. So this is an important property too. And in some cases, we also will evaluate in vivo properties. For example, if we have a few candidate formulations and we want to know, okay, which formulation has the best bioavailability in an in vivo model, then we can set up, for example, rat and mouse experiments. And you see here some examples of profiles. The same API, just a different vehicle where the API is solubilized. And you see that the PK profiles in rat can be very highly different. So from this, for example, you have a very high bioavailability from this in vitro formulation, and then for the green one, for example, capriol formulation, we have a low bioavailability. So the choice of the excipient, the choice of the vehicle is very important in some cases for the bioavailability. So now I will zoom a little bit more on the IV2Sol. So what is IV2Sol? Well, it is, in fact, a reversed dissolution testing. So we will start with a number of candidate formulations. Let's say the API solubilized in maybe 10 or 20 different vehicles. So we are talking about solutions, and so we solubilize the API in vehicles. But what can happen in a gastrointestinal system, and that is unwanted, that is that when the formulation comes into the gastric or intestinal environment, that the API precipitates. And so we need to avoid this, because if the API precipitates, it is probably not bioavailable anymore. So what we will do, we will dilute these candidate formulations in relevant media, like simulated intestinal fluid, simulated gastric fluid, and we will do that on a very small scale. So we need only very limited amount of API to do this experiment. We will incubate these plates. So these plates contain all a number of the vehicle, so the candidate formulation, plus the aqueous simulated fluid. Then we incubate at 37 degrees, and we follow a response. We follow the optical density in these wells using an optical density reader. So what happens? If nothing happens, if the well remains clear, like in this example here, that means that even after dilution of the formulation, the API stays solubilized. So that would be a good candidate. But we have other wells like this one here. This is a candidate formulation giving precipitation that can happen immediately or can take a time before precipitation takes place. This is not a suitable candidate. So I give you some example curves. So what you see here is optical density curves in function of time. So we follow this test for 1 hour, 2 hours, and then we follow the optical density. And you can see here some vehicles like the red one here. The well remains clear, so no precipitation at all over a period of 120 minutes. So this would be a very favorable candidate for further selections. So this was a formulation solubilized in cyclodextrins, but also some other formulations like this one and this one's here, they also show only a limited precipitation, so they are also suitable. The ones here on top with, for example, here the cremophore formulation or transcutol formulations for this API, they show quite fast precipitation, so we would not withhold these ones. We would say these are not appropriate formulations, and we would continue the development, stability testing and so on with these formulations. So this would be, the system is used in fact to rank formulations, which are good ones, which are bad ones. And so it's not an absolute technique, because of course this platform only looks into the terms of solubility. And so if the permeability is an issue, then with this platform you will not identify that. So this is the limitation. It's only focused on solubility. Now some words about analytical development. So the laboratory, the analytical laboratory at Pharmavize is a development lab. So what do we do there in the lab? Well, we will put a lot of effort on impurities determination. So often we have to develop a suitable HPLC or UPLC method in order to identify the degradation products that we pick up during the stability study. So a lot of efforts there on a suitable method development for impurity determination. We prefer to use UPLC methods rather than HPLC, because the benefit of UPLC is increased sensitivity, increased speed, so you can do more trials, more experiments in a working day. So everything goes faster and better with this UPLC. Then for the method development, well, we have a very good knowledge of the regulatory framework, so that means that we will use an appropriate validation versus the development stage. For a phase 1 study, it is not yet needed that you apply a fully ICH validated analytical method, so you can do, let's say, a basic validation that will be sufficient for an early study. Of course, if you go to later phases, you will have to upgrade your validation standard and do, for example, also robustness testing, the tests that you will need to do a full-fledged validation. We will also develop always stability indicating methods, so stress testing of the drug product and putting these stressed samples on the candidate analytical method is always part of method development at Pharmavize. And the way we develop methods, preferably, we start from the API method, which is given to Pharmavize by the API supplier, and then we start modifying this method to make this method suitable for the drug product. Also for dissolution methods, so for the solid dosage forms, dissolution is a very important characteristic. We are also known, and our lab is well suited to develop suitable methods, and the important word here for dissolution methods, it needs to have sufficient discriminative power. That means that your dissolution method should reflect as good as possible the situation in vivo. That means a well bioavailable formulation should have a good dissolution profile. A lower bioavailable formulation should have preferably a lower dissolution profile. So this is what we call the discriminative power. The method should be able to discriminate between good and bad formulations. And then some words about GMP manufacturing. So we have clean rooms, small clean rooms, to accommodate small batch manufacturing. And what is typically for clinical manufacturing? Well, you need a high degree of flexibility, because you cannot compare it with commercial GMP manufacturing. Often the API that we use is not always representative. It could happen we do, for example, the development with a non-GMP API batch, and then we start in the GMP area with a GMP batch of API, and suddenly we see that the solubility properties, for example, or the impurity profile of the API GMP batch is not the same as before. So you need to cope with that kind of problems that can happen during GMP activities, that you see different things as you have seen during the development. Also, the production processes are still under development, so we are working with not yet validated processes, because it's the first time we manufacture in the GMP, so you also need to be to cope with that, and you can have surprises where you have to cope with. Also, what can happen is that the clinical study design can change during the process. It can happen that we start, for example, with the preparation of the manufacturing of a GMP batch for 50 milligram dose, and that a few weeks before the clinical study, the customer would like also to have a 100 milligram dose. So then you need to be flexible in order to cope with these requests. So this means that we will apply a non-routine approach, so every project is different. It's not a repetitive business. It's always, for every manufacturing, is new compounds, new formulations. And we apply, for this we applied staged automation. That means that for very small batches, sometimes we only make capsule batches, for example, of 10 capsules. If it's a very limited study with only a few patients, then it can be that we make batches of 10 capsules. So this is a very low amount, and if we have low amounts, lower than 5,000, we will often use manual filling technologies. If the quantities are higher, we go to semi-automatic filling, and really for the higher batches, for example, phase 2, and when we go to phase 2 studies, we often need 20,000 or even 100,000, then we will go to automatic filling processes. And here you see, in a nutshell, a development project for a clinical phase 1 project. Well, first of all, it will take, on average, because it will depend from project to project, it will take about 10 months of work. So we will start here in the beginning with solubility excipient compatibility tests. So this takes about 2 months. Then we will make some formulation concepts. Can be 5 concepts, 10 concepts, and these concepts are placed on a concept stability program. So we will run, for example, 1 month stability studies at 40 degrees, accelerated conditions, and then we will select the suitable formulations, and if we do not have a suitable formulation, we restart with a second campaign of candidate formulation, so optimization of the formulations. And when we optimize formulations, we do another stability study of 1 month at 40 degrees. Along with this stability testing, we are busy with the analytical development. So this goes along with this activity. So in the beginning, we start with, let's say, an initial method, but during the development, we will further fine-tune the method and validate the method. And then when we have here, after the screening stability, we will then make our final selection. This will be the concept for the clinical study. Well, then we will go to a non-GMP representative batch manufacturing. This means that we will make the batch, but still using the GMP equipment, using the GMP type of facility, but producing it under a non-GMP quality. Why do we do that? Well, it has many purposes. First of all, it is needed to fine-tune the process, to find the right operational settings. It is also needed to gain time, and so because we will put this batch on a stability program, and this stability program will be used for clinical, for the regulatory authorities, and also it's a general repetition of the GMP. So if you can do a non-GMP representative batch, the risks at the GMP level will be lower. So this non-GMP representative batch is placed on a stability program. This can be 1 month, but if we have time enough, we would prefer to go for 3 months stability data. Along with this stability study, we are already working on the regulatory, so on the writing of the IMPD or the IND. And then when the stability data comes available, then we will enter the stability data in the IMPD and submit to the authority. Along with that, we are then already preparing the GMP batch manufacturing. So here you have the non-GMP, which goes on the stability, and then you have the GMP batch, which is then used for the clinic. So in this way, by using this non-GMP batch, we gain time because we do not have to wait until the GMP batch is ready before we can start with stability work. And then finally, we have to do the quality control and the batch release. So here, as at the end of my presentation, I want to give you some take-home messages. So first of all, the complexity of CMC development processes is mainly determined by the physicochemical characteristic of the active substance. Also, the regulatory framework is important and the number of parties involved. So one of the, let's say, business model of Pharmavize is to have all these different services going from formulation, analytical, stability, GMP, early GMP manufacturing under one roof. So this gives that everything can go smoother and that you do not have a lot of different parties involved, so it can go faster. What our recommendation would be for the early clinical trials, avoid complexity. If, for example, a customer would like to go in the phase 1 study with a very fancy formulation, very fancy multi-layer tablet, for example, this is not, let's say, the ideal situation. So for a phase 1 study, we say make it as simple as possible. If a simple solution and the API and water, for example, works, well, go forward with that. Don't make it more complicated than needed. Also for the powder, just take the API, blend it with some filler, a lubricant glidant, and put it in a capsule. So this is to avoid complexity, make it easy, and then you have less troubles. Later, of course, you can then further make more advanced formulations, but in the early trials, also to gain a lot of time, we try to keep it as simple as possible. And the early GMP manufacturing requires a dedicated, non-routine approach. And so a contract manufacturer busy with commercial manufacturing is often not well installed, not well suited to do this, let's say, small clinical, early clinical manufacturing. So we have found this as a niche for Pharmavize to just focus on this kind of small-scale early GMP batches. And also last but not least, the speed of development is important. I said it in the beginning of my presentation. Once a customer has decided to move forward, we must be ready to go fast, step by step, in an accelerated mode. If you want to talk further with us, we are here in this hall in booth 1F61, and you are we would be happy to have you in our booth later today. Thank you very much.